Welcome to a very short introduction to laminins. First, some context. This is a skin section where we've stained the nuclei in blue, fibronectin in green, and our favourite protein, laminin alpha 3 in red. And what you're seeing are layers of epithelial cells sitting on top of an extracellular matrix at a specialised region of matrix termed the basement membrane. And in that basement membrane we find proteins like collagen 4, but also laminins. So the laminins are a multi-gene family derived from single ancestral origins. And they're broken down into 5 alpha chain, 4 beta chain, and 3 gamma chains in humans. And each laminin protein is a heterotrimer consisting of 1 alpha chain, 1 beta chain, and 1 gamma chain. And these trimers form via laminin coil domains, which are present in them all. At the end terminus of some, but not all, are domains called LN domains, and these are involved in laminin to laminin interactions or laminin polymerization, as we'll see later. Whereas at the C terminus of the alpha chain are LG domains, and these are involved in cell receptor binding. So laminins form heterotrimers, and we name the heterotrimers based on their chain composition. So laminin alpha 5 beta 1 gamma 1 gets known as laminin 511, whereas laminin 332 is alpha 3 beta 3 gamma 2. Once assembled, the laminin heterotrimers get deposited from the cell, but they stay cell associated by binding to cell surface receptors. There are 2 major integrin-based adhesive devices formed in epithelial cells. On the left-hand side is the focal adhesion, which involves alpha 3 beta 1, a host of other proteins, and ultimately recruits the actin cytoskeleton and is involved in driving the forward migration of cells. Whereas in contrast, on the right-hand side is the hemidesmosome, which involves alpha 6 beta 4 integrin and a number of binding partners which associate with the intermediate filament cytoskeleton are involved more in cell-to-matrix adhesion. There is a large amount of crosstalk between these 2 different complexes, and the hemidesmosome has also now been shown to be involved in regulating the steering or the directionality of migration. However, the major role of the hemidesmosome and the best studied role is exemplified by the human genetic disease epidermolysis bullosa, where mutations to any of the components of the hemidesmosome, including the laminins, lead to skin fragility and blistering. In cells, focal contacts look exactly like they sound, discrete puncta towards the periphery of the cells, whereas the hemidesmosome is a more like a rose-like array, rosette type pattern. It's very pretty. In addition to their cell surface receptors, the lamins also bind to themselves to form a network, and they do this by using their LN domains. The current model is an alpha beta gamma LN domain needs to form a ternary node. Once the polymer is formed, other binding proteins such as nitrogens and perlicans can bind, and these can associate with things like collagen 4 in the basement membrane. The way that the lamins are deposited by the cells actually depends on which type of cell that you're looking at. So skin cells do a rose-like array, whereas you get more fibrillar arrays in alveolar epithelial cells and more punctate distribution in endothelial cells. And that reflects largely the type of cell behaviour that's supported by these proteins. So next I'd like to introduce the LaNts, which is our own little babies. So when I was studying one of these rare genetic diseases, we noticed that there was a number of transcripts derived by alternate splicing from the end terminus, the 5' end of laminin encoding genes. We call these now the laminin N-terminal proteins, or LaNts. And they're named in the same sort of way, so based on the chain that they're derived from. Most of our studies are focused on LaNt alpha 3-1, which contains an LN domain and a short stretch of LE repeats, but no LCC domain, so cannot actually form a trimer with laminins. However, they do locate to the same location in the skin, toward the basement membrane, and they decorate regions where laminin 332 is present. In the only study published to date, knockdown of LaNt alpha 3-1 in skin cells was performed, and you lose those peripheral puncta that decorate those regions of laminin 332 in the cells. And one of the most striking phenotypes is that in the cells that don't have this LaNt alpha 3-1 protein, the cells fail to close a scratch wound in vitro. So this protein seems to be involved in cell migration. So we encourage you to read that initial paper for loads more details on these proteins, and look out for our next one coming soon. Finally, use the link below to check out our website and find out about the lab.